Hello everybody, this is my video. My name is Wes Butler and this is for Genetics 375 at BYU Idaho. The directions for this video is just 2 to 3 minutes, briefly go over how I went through these problems. Let's start off with problem 1. It says, suppose you chose the following cross, AA, BB, yada yada yada. Pretty much what I do with my problems, I'll show you here, is, I don't know if that's backwards or not. Still trying to figure out how to do the screen share on this live stream. So I don't do the forked method. I do just the possibilities, and if, say, it's like a 1/4 or a 1/2 or a 1/4, and that way I don't have to make a new fork for each one. So I can just write all the genetic code on there, and then I say I refer to this AA up top. I say, oh, what is that one? That's one's 1/2, and then I just get all the fractions from all those different ones, and then I just multiply them all together, and that gives me the answer. That's how I did the first one. For number C, I didn't put in a number. It's a 0 because the lowercase dd, as you can see, did not appear on there. And that was because one of them had 2 uppercase D's for those alleles. So that's how I did number 1. And then let's go to number 2 here. It says fill in the blanks. Paired homologous chromosomes are sometimes called tetrads or bivalents. That was just from the PowerPoint. The structure that connects the centromere to the microtubules of the mitotic spindle is called the kinetochore. And this is C, and some of these, like the ones in C, I wasn't as totally confident on, but here it is. Diploid fruit fly. The first answer was 16. That's what I got. The second answer is 8. The D is Punnett square, E is null, and hydromorphic. Mendel's independent assortment involves 2 traits, while the law of segregation involves a single trait. Talking about number 3, 3A, I didn't really understand, and given the limited time right now, all I put is this. So, I don't know how else I would arrange those. I felt like they should all go together. B did make more sense at all the different variations that could occur at the conclusion of meiosis 2. So there would be just a single one in each one. That's those possible variations. And then sister chromatids found per cell in this organism during metaphase 1, 6, and chromosomes in organism during metaphase 2, 1.5. That was also kind of a question mark for me. Didn't make sense, but don't have time to go over it right now. And then number 4, I feel like was the same thing, but I feel like I understand that one a little bit better. Don't have too much to show for that one. Number 5 was pretty good. This was my work for number 5. So as you can see here, I did this again, where I made those little boxes. I didn't do the fork tail, but it's pretty much the fork tail. I just didn't want to write everything so many times. So then I have the fractions next to how many times each one can occur. And I get those fractions from these little Punnett squares. Out of 4, the double capital T is going to occur once. So that's why that is a 1 out of 4. And that's how they do it for the fork tail method. And that's how I did it for this as well. And then I can just, I didn't even bother to write as much on this one. I just wrote the fractions that I needed for each one, multiplied those together with my calculator and got an answer. And that was it. But good luck with the semester, guys.